Waleed Faisal, and I'm a project manager and career fit fellow in the School of Pharmacy. I graduated from School of Pharmacy in 1997, and then my wife moved to start her master in a different city. So I follow her, and I finished my master in 2002 in pharmaceutics. Then I look for international PhD scholarship, and I finally landed in UCC in 2005, and I awarded my PhD in pharmaceutics from School of Pharmacy in 2009. Then I work as a postdoc in the School of Pharmacy. And then I moved to Mania University and I work as a lecturer in pharmaceutics. And in 2017, I applied for a Marie Curie Fellowship to achieve the skills and technical experience to build my own research team. And the project was about the microneedles. And as an outcome of this fellowship, I start to investigate the opportunity to create or to start up a company around this technology, and to transfer the innovations and which developed in UCC to industry. During this career, I built an experience in designing different multidisciplinary research projects in the pharmaceutical formulations. And I published a number of papers, and I am inventor in 2 patents in this area. And I'm currently leading a team in the pharmaceutical development and commercialization of the microneedle technology. Okay, so this is a quick introduction, and then we'll go to the project today. So before we start, if you don't mind, just if you can go to slido.com in your phone or your laptop or your desktop, and just describe yourself, if you know, are you a student or researcher or academic, so you know, to start to talk about the project. So we'll give it 1 more minute. Okay, so I can see that, you know, most of the audience are from the undergraduate students. So I will not go so deep in the technical issues or technical analysis about microneedles. Okay, so the first thing to start, if you go again to Slido, do you know what's your largest organ in your body? You know, we have some internal and external organs, but which one is the largest? Yeah, that's good. So yeah, the skin is the largest organ in your body, and it's the one we're wearing all the time from outside, and it's about 3.6 kilogram, and the size is 2 square meters. And because it's the largest organ in our body, always the pharmaceutical formulators thinking about targeting the drug through this delivery system, also, sorry, through this route of administration. But the one issue of the tissue, the skin tissue, which make a challenge for using this route of administration is the anatomy structure of the skin. As most of you may know, that the structure is mainly two parts, epidermis, and dermis, and hypodermis. And the outermost layer of the skin is the stratum corneum. It's a dead cells layer, which is called the stratum corneum. And this layer, because it's dead in nature, so it's protecting our body from any environmental condition. It protects you from the light, from heat, from the virus, from bacteria, and also nothing coming out of the body. So this layer works as a protection layer for your body. At the same time, it's a barrier for any delivery of medication from any pharmaceutical formulation. So in pharmacy, we start thinking about some topical drug delivery system through the skin, and we think about the creams or ointments which is applied to the skin. But we end up that very little amount of this drug can penetrate the skin or can diffuse through the skin and reach intradermal. And this is again because of this dead layer. Then we thought if we can work with something which can attach to the skin, which is connected to the skin for longer time, like the topical patches, which you can leave it in your skin for maybe hours or days. But again, the amount of the drug which go through the skin was still very little, and the reason is the same layer. Then the formulator starts to think if we can bypass this layer, then we can deliver much higher dose of medication internally. And the idea was coming with the hypodermic needles. So you have the needle, it goes through the skin, so we bypass the dead layer and deliver the medication internally with a high dose, which give you a more accurate dose, intramuscular or intravenous or subcutaneous. But always in an issue with the nerve endings. The needles will once go through the skin, touching the nerve ending, then it causes pain. Okay, and if you think about the hypodermic needles, which are a very, very old technology, and if you try to find out, do you know how old this syringe needles? Can you imagine how old is it? Yeah, the microneedle technology, sorry, the syringe and needle technology is very old technology. It's more than 170 years old, and it was first invented in Scotland. And this technology has many shortcomings with the use of it. The first shortcoming is the fear. As we mentioned before, it once is touching the nerve endings, it causes pain. That's why people start feeling pain and don't like to use injection. And this pain will increase and reach to be a needle phobia. And it's mostly about 20% of the world population have this phobia, where they are trying to avoid taking injections. And myself, one of these people who have this phobia of injection. This is one of the shortcomings of using the needles and syringe. Another problem with using with the syringe needles is the needle stick injuries. As you can see here, when you finish using the needle, the waste needles here, it may cause contamination or cross-contamination. And WHO suggests that about 1.3 million persons every year die because of this cross-contamination of the needle stick. Another problem with using the needles and syringe is something we may heard last couple of months when they have the COVID vaccines from Pfizer. And they mentioned they need to transport it under -70°. And this is what we call the cold chain transportation. Most of the, or maybe all of the injections need to be transported from the time of production to the place of application in a cold chain or in refrigerators or in a low temperature. And the WHO mentioned that half of the vaccines all over the world considered to be not working properly because of the lack or insufficient cold chain transportation. So because of these three main problems with using needles and syringe, we here in UCC School of Pharmacy, we start to think how we can tackle these three problems in the application of needles. Okay, and then the idea is back again to the same photo. We think about a technology which is similar to the needles, but you need it long enough so it can bypass the layer of the dead layer of stratum corneum, which is about 0.2 millimeter. And at the same time, you need it short enough so it will not touch the nerve endings. So you bypass the stratum corneum, the dead layer, and you didn't touch the nerves, so will not cause the pain. At the same time, we're looking for a technology which is solid. So if it's solid, that means it will not need for a cold chain transportation, because most of the cold chain transportation mainly for the liquid or solutions. Also, if this technology can dissolve inside the body, so we will not get any waste needles. So we're looking for something dissolving the body, long enough to bypass the layer of the skin, short enough not touching the nerve endings, and same time it's solid state, so we don't need for the cold chain transportation. And this is technology, what we call it, the microneedles. And you can see it here in the picture, the microneedle size, from the name, it's a micro-sized needles. This is the normal needle which we use, and this is the tiny structure of microneedles. And you can see here a patch, this is called array patch. A batch of microneedles, which is 1 centimeter square, can accommodate up to hundreds of these small microneedles. At the same time, if we go to the millimeter scale, you can see the needles here, how it's sharp, very short. It's about 500 micron or half millimeter. It's strong, and the base is about 250 to 300 micrometer. So you can imagine how small the size of these microneedles, how sharp it is to deliver the medication. When you look to the structure of microneedles here, it has two main parts: the base of the microneedle and the needle itself, the needle body. Okay. And the microneedle delivery system is not a new technology. It is a bit old one. It's first mentioned in literature about 1980s when you talk about microneedle and use it in a cosmetic application. And from that time till today, there is a lot of designs of microneedles coming for the application in the for drug delivery. The first design of microneedles called solid microneedles. Meaning of solid microneedle, it means it's the base and the needle body itself are made of metals. When you apply it to the skin, it penetrates the skin and make an hole in the skin, and you can see it here in the video. So when you apply it to the skin, you make a hole in the skin, and then you can apply a layer of creams or ointment which has a chance to goes diffuse through these holes to reach the tissue intradermally. And this is the technology. The problem with this first design of microneedles is that your skin or everybody's skin are elastic, which means after the application, after having these holes, it easily closes down very quickly, which make the drug diffusion are fluctuated for how long these holes will stay. So this fluctuation makes the application for this technology for drug delivery are not suitable. But it's okay if we can use it for the cosmetics or beauty centers. Then the second design of microneedles is called coated microneedle. And from the name of coated microneedle, it's the same structure from the solid microneedle. It's a base and needle body, but then we coat the needle with a thin layer of medications. When you apply it to the skin, when you insert it to the skin, as you can see here in the video, you deliver the coat inside the skin, and then you take out the needle. And then this layer, this coat, starts to dissolve to deliver a constant or a same amount of drug every time. The problem with this technology is that the coat layer is very tiny, it's very thin, and the needle is small. So the amount of the drug which you can deliver with this method is a very small amount, which limited the application of this new design. Then the formulator think about a different way where we make the needle from a base and body from the metal, but the base become a reservoir for the medication. It's like a stock drug. When you insert it to the skin, the needle itself has a hollow inside. Every needle has a tiny hollow, and the drug then can diffuse from the reservoir through the hollow to reach inside intradermally. Okay, this happens with the osmotic differences, osmotic pressure differences. This was very good because you can now deliver a higher amount of medication from the reservoir. The problem was the needle hollow, this hollow, was very small. It's a micron scale. When you insert it to the skin, the tissue and blood cells and the tissue cells can easily block these hollows and make the delivery or the transfer of the drug very slow, maybe stop. The formulators start to think another application for this. They said, instead of transferring or delivering the medication to the body, we can collect the fluids from the skin through this hollow. So we collect fluids and blood from this hollow to outside, and then we can measure something like measure the glucose in your body with something simple. All these designs now have an issue with the all made of metals, and sometimes they made it from ceramic. When you insert it in the skin, because the size is very tiny, it's in microns, less than 0.5 millimeter, it can easily poke in the skin, which make a serious health problem. Then we have another design for the microneedles, which is the most used design of microneedles nowadays, which we call it the dissolving microneedles. In this, we have the substrate, we have the base, and then we have the needle body, which made out of a mixture of polymer and the medication. When you apply it to the skin, go in, and then the polymer start to dissolve inside the skin and releasing the medication to give the effect internally. The problem with this technology was to get the polymer to form the needle body. you need more than 90% of the structure of the needle is just polymer. So to give the strength and the structure of needle. And only we end up with less than 10% of the size of needle is the medications, which means that very little amount of the drug can be delivered. The second problem was these polymers are already regarded as safe. So they are safe to be used in human, but all the details about the safety is for oral use, not for skin use, not for dermal use. And you need a lot of clinical trials to prove that they are safe to use intradermally for long term, which will make extra cost for any investors to work with this technology. Then we try here in our team to tackle this problem. We need to increase the drug loading in the needles. Same times, we need to get rid of the excipient or the polymer, so we don't need to do extra clinical trials, and so we're decreasing the cost of production. These are the three main challenges of this technology, which we thought is good if we reach a solution for them. At this stage, we start to think about the glass. So if you know the glass, how we made the glass, it's just sand particles, which we heated up to the melting point, and then we have a melt, and then we can shape this melt to form a glass sheet or a bottle or eyeglasses or any kind of glass products. So we're just using the heat to produce the glass products. And we thought if the medications can be done or can treat it same way, if we heated the medication particle or drug particles at the melting point, and then we have a melt of the particles of medication, then we can shape it in a structure of tiny microneedles. Then you will get a needles which is completely 100% made of medication. And same time, these needles are strong enough to penetrate the skin once it's cold and became solid. So we start to look for medication which has these characters, has something we call it glass transformation. It can be transformed to a glass status when it's melted. So when it's melted and cooled down, it will back to be, instead of powder, it will form a structure of glass, which is shiny and strong enough to penetrate the skin. And this is what we call it glass transition. And this technology, we patented this technology last year with an international patent. So we identified a therapeutic commercial opportunity to have a new microneedle array patch, which to localize the medication intradermally with 100% medication, so the maximum drug loading, no polymer, so there is no need to do any clinical trials. Convenient for patient because these needles will not touch the nerve endings, so the patient will never feel any pain when they apply it in the skin. Less cost in production because it's simple heating production methods without using any clinical studies for polymer, and is patent under our name. And this comes with the technology, the microneedle array patch technology in School of Pharmacy. This technology claims that we can structure or manufacture the microneedle with 100% medication and with a novel melt process technique. To date, we identified 18 different drug molecules according to their physical and chemical characteristics, which can be melted and manufactured with this technology to form a sharp microneedle which can penetrate the skin. So this technology was expanded to be a platform technology for treatment of different diseases like antifungals, corticosteroids, calcineurin inhibitors for allergy and eczema, anti-inflammatory, pain managements, antibiotics, and cancer treatment. Then at this stage, we search for a feedback from the key opinion leaders in industry and industrial stakeholders about what's the opportunity for this technology in the marketing. And we got a commercial case feasibility study from Enterprise Island to search and look for the feedback from the stakeholders. And the industry stakeholders confirm that there is unmet need in the market for such technology. And they identified that the nail fungal infection is the best route or the best market to start with. Then we talk with the clinicians who are interested or are very interested in idea of localize the treatment. Sorry? Okay. We start to talk with clinicians who are very interested in the idea of localizing the treatment of microneedles, and they are interested in to use it in the nail fungal infections. Then with the patient feedback, they are very interested with a new technology with less pain and same time less side effects when treating the antifungals. So why antifungal? Why they choose antifungals as the best route to start with? Nail fungal infection, or we call it onychomycosis, it's a health substantial disease. It affects about 10% of the whole population in the world. It's a chronic and resistant to treatment. And the most effective treatment is the systemic treatment with tablets. But with the systemic treatment, you need to use a very high dose of medication for a very long time, about 3 to 6 months, and sometimes you reach for 1 year. Hence, you have a lot of side effects, a lot of drug-drug interaction, and same time, you get very little patient adherence for this long time. Then we have another treatment, which is a topical treatment as a treatment for fungal infections. This one is the biggest market for antifungal, but it has less efficacy because of the skin barrier and the nail barrier. But it's less side effects and easily to buy it from over the counter. And recently we have the new technology of laser treatment and electrophoresis, but this kind of things is very, very expensive and need health professional all the time. Although all of this kind of treatments, there is about 16 to 25% of patients, they will never achieve the full curement. And the recurrence rate is about 10 to 50% in patients. If we look to commercial side, the market of the nail antifungal is about $4.6 billion in 2017. And it has a growth rate, it will witness a growth rate of 6.7% from 2018 to 2026. And the main reason for this is the overpopulation, especially for the growth of overpopulation, especially in the geriatric patient or elderly patient who are more infected with nail fungal infections. When we talk about the site of application where we can apply this microneedle technology, in this case for the nail fungal infection, we apply it here in the area we call it the nail fold, which is the skin attached to the end of the nail. And this area of skin composed of nail root and the nail matrix here, where is the tissue the fungi are resided during infection. So we apply the needles here, which go inside here and start to dissolve to attack the fungi. So what we did in our work, we start to screen the different potential antifungal agents and to find which one is best to start, and we get a decision, is it the best antifungal to start with a compound called itraconazole. And itraconazole has the ability to be fabricated with the melt method. And this is how it looks, the needle itself. You can see how is it sharp, very strong and shiny like glass. And here with the electron microscope, you can see the sharpness of the needle. And you see the 8-sided structure of the needle. And you can see this needle is 0.5 millimeter height and 1/4 millimeter diameter. This is how small is the needle. And this sharpness and the strength of the microneedle will permit the insertion through the skin layer to deliver the medication. Then when we apply it in ex vivo, when we apply it to the animal skin, to the surface of animal skin, we can see easily here the holes which created by the needles. And then the needles start to dissolve and has the ability to deliver the medication intradermally in the skin. Then we start to look for this microneedle, to look for the chemical stability, the physical stability. So we look for the drug content, how much of the drug in each needle. The texture analyzer to check the strength of each needle. Is it strong enough to go through the skin? And then we get imaging of the needle, like we see in the last slide, how it looks from every angle. Then we look for the chemical stability. So we use a different technology or different analysis method to prove that the drug or the microneedles or the melt we're using are stable before and after melting. Then we look for the efficacy. So we try to see the drug itself before melt is known as effective for treatment of fungi. But after the melting, after heating, is it still stable and effective in treating the antifungi? It still has the ability to kill the fungi? And we prove this the efficacy before and after melting. I will not go too much through this chemical and studies, I'll not bother you about it, but we did a lot of work to study the different technologies like DSC, XRD, NMR, LC-mass, the minimum inhibition concentration to see which fungi they can kill. We did a stability study to see if there is any degradation happens. All this kind of study to prove that this technology is working. What we're doing now, we're trying to direct the proving the platform concept. We're trying to work in proof of concept of the platform with different molecules. So we have a pipeline for the future application. So we're trying to identify what's the best market to follow the itraconazole, to follow the antifungals application. And then we work in the in vivo technology or in vivo technical development, which we're trying to see how this molecule, how the microneedle work in the animals, is the efficacy and the safety in animals before we go into clinical trials in humans. We're also seeking the potential manufacturing and clinical partners to collaborate in this microneedle technology in future. So this is mostly the work we've done. This project has funded through Marie Curie projects in 2018. And also we have a fund from Enterprise Island, the case feasibility study in 2019. And then we also have funding from Enterprise Island Commercialization Fund in 2020, which the project is working now. And that's all. And I would like to thank the group working with me, Professor Abaina Green, which is the technical advisor in the group, and Dr. Dennis McDade, he's the industrial advisor. We have Nicole Martinelli in the group. She is a Marie Curie projects manager. Dr. Fatma Farag from the technicians in the School of Pharmacy. We have Kevin Dalton. He works in the technology transfer office for work with the patents issue. And we have Claire Walsh. She is a commercialization consultant. And thank you all for listening. And I hope you enjoy it. And I'm happy to take any question. And thank you.
Thank you so much, Waleed. That was a really interesting STEMinar, and your research sounds really, really promising. So I'm looking forward to seeing it on the market. So we'll start off with a Q&A session. So just if anyone here wants to ask any questions, please feel free to send them into the chat, or else if you want to turn on your mics and ask them, that's no problem. So I have a few questions here anyway, and so we can start really from the beginning. So where would you say that like your whole interest in pharmacy stemmed from? So, you know, what encouraged you to start studying that?
Okay, that's a good point. It's very early in my life, I wasn't clear, when I finished my leaving cert, I was thinking about doing medicine or something near to this area. And also I was interested in I love math all the time, so I was thinking, oh, should I do something related to the math? But then I landed with find the pharmacy. It has a lot of medicine part, and same time you have a lot of commercial works because you work in the market, and so I feel it's more near to me. That's why I start with the pharmacy. Then when I start my master degree, I start, I feel I'm more related to research. I like to do more research. I like to have a group working with me, I have a lot of ideas. I like to investigate it. That's it.
Thanks, Solly. That's really interesting, especially to see like, how you started off as well and how that's developed into microneedles now today. So in terms of your research, the microneedles, what would you say has been the biggest challenge that you've faced with it, and how have you overcome that?
Okay, the first challenge was, when we start, how we can, before we have this idea of the melting method, it's always about the challenge, not just for us, for everyone working in the area of microneedle, is how you increase the amount. The needle itself is very tiny, very small, and the amount of the drug you can deliver is very, very, very small. Okay, and to increase this amount to reach a maximum, so you can use it for treatment. That was a big challenge. How to be sure that you have enough amount of medication in the needle patch. And this was the first challenge, which made took a lot of time till we find out a way to increase the loading to 100%. Another challenge with microneedles was, okay, when you apply it in the skin, the idea always, we need to localize the treatment in the skin, in the site of infection. Okay, but if you apply it and your drug dissolve and reach the systemic circulation, then it will distribute it everywhere in your body. Then the dose or the concentration of the drug will be very small if it's went everywhere in your body. So that challenge was how we can localize. So once you apply the microneedle in the skin, we need it to stay as long, as much as you can in the same place. Don't go anywhere. So we start to think about the molecules which have this ability to stay in the skin or to stay in the lipid of the skin. All the skin, the layer, they have lipids in it. So we thought about, if the molecule is lipophilic, he loves the lipids, then when you apply it, he will dissolve in the lipids, he will stay, he will not go to circulation because your blood is water. So he will love to stay in the skin more than it. Eventually, it will go to the systemic circulation, but if it take more time in the skin, then he can treat the infection in the skin, and you can get a good concentration for a good while. And this is why we start to think only for molecules which has a higher affinity to the lipids, which we call it high log B drugs molecules.
Yeah, that's really interesting. And I guess it just highlights the importance of having to think outside the box when it comes to research. You have to like think of all these questions and ways to go around them. That's really good. Yeah. So what part of your microneedle research do you think you enjoyed the most?
What part of microneedle?
Yeah, the whole journey from the start to now.
The commercialization part.
Really?
when I start, I start to think about, about 1 year and a half now. We start, because as a start, it is when you work as a researcher, you think as a scientist, okay, which is very clear. But when you think about commercialization, everything changes. You may have a very good idea, one of the best ideas in the world, but there is no application, or there is no investor will come for this, or you may have something very simple but easy to tackle the commercialization. So it's a different world, and it's research just for research is different than research which is targeting commercialization. So you think about what market need first, not what you know or what you can do. What's the market need? What's he looking for? And then, okay, it's easy to go to an investor and tell him about microneedle. Oh, that's a very nice idea. Then you ask him, could you invest in this? That's different. Oh, you'll start thinking, okay, anyone need it? What's the back, what I'll get, what's in it for me? So when you think differently, you start to think, about your research in a completely different way. But I like how, I start to do a lot of studying in commercialization and business management. Yeah.
Yeah, it's like you really have to put on your innovation cap as well as your entrepreneurship cap, really. Yeah. As you said, like if you could have a great technology, but you really just need to look for the gaps in the market to see, who it'll benefit. Yeah, it's really good. We've actually a question in the chat from Jessica, and she's asking, do you think microneedles could be used for the administration of vaccines in the future?
Yeah, that's the main reason why they start the technology of microneedles. The first application for microneedle was the vaccines. Okay, they tried because the problem with vaccines is, we talk about the cold chain transportation. A lot of vaccine you have to transfer it, especially to the Middle East or the Africa or this country, which is very hot. With the lack of cold chain, you end up with the vaccine was not working. Okay, that's why they're trying to get more coverage of the vaccines in these countries, and the first solution they're thinking about is microneedles. And I think Bill Gates and Melinda organization, they investing a lot of money in this area about microneedles and vaccines. I think they have a couple of clinical trials in phase 1 or phase 2 about flu vaccines. Nothing yet for COVID, but I think there is a lot of work in flu vaccines with microneedles.
Yeah, it certainly does sound very promising.
Yeah, and I have to say, the microneedle, there is nothing in the market in microneedle. The nearest group in US, a company called Zusano, I think, they are in phase 3 clinical trials. So maybe within a year or two, they will reach the market, and they work with migraine. People who has a migraine, they have a very good drugs, medication in the market, which is working very well for these people. But the issue was, once you take the tablet, you need about 2 hours to get rid of the migraine and the pain. And because the pain is very hard, the patient will go for microneedle, because microneedles is faster in the absorption. So you will get the efficacy within half an hour. And they thought because of this time differences between half an hour and 2 hours, they will get a good market share for treatment of migraine.
Yeah, and I like the migraine market. You know, there's so many people out there that are suffer from migraines, and to see that there's going to be a novel product like this on the market for them, it's
Yeah.
very interesting. Um, what would you say that a typical day in the lab, you know, working on the microneedle research looks like? You know, do you have a balance between, you know, being within the lab and doing experiments and with the office work as well when it comes to write-ups and things like
It's
that?
different from stage to stage, when we start the project, there is a lot of lab work. So most of the time, we start, we have the plan of the work, what's the deliverable, you have the milestones, and then the dates, and then you start working. You have a lot of lab work. But once we finish, we get a patent, this technology, and we start to think about application, going to the market, then we get more time for fund applications. So a lot of work, writing applications, a lot of management, because I have a team now working with me. So management in the team, it take more time. Meetings, with the COVID and lots of meeting now. Less lab work because there is restriction in the number of people in the lab. But I would say it depends on what stage of you in the research. If you're starting your career in research, of course, most of the time will be lab work. As you're going in the research, with time you start to getting more time for just administration work and management work more than research. Okay.
Yeah, that's really interesting. Thank you. And you know, for anyone here who say has is plans on doing research or innovation in the future, what advice would you have for them if to apply for funding for the research?
Your question again?
And what advice would you have for, you know, future researchers here in the room for when they have to apply for funding for their research projects, like you had to do, say, from Enterprise Ireland and
I
things like
think,
that?
I feel, the Enterprise Island, for example, which is a very good place to apply for funding. If you have an idea, you feel there is an application for this, there is a future for this in commercialization, just go for Enterprise Island. You have a very good rate for applying for, for success rate. You have a good success rate if you have a good idea. They have a supporting, so you will have your own commercialization specialist who will sit down with you maybe every week to discuss the idea and find the way of writing. They are very helpful. Also in UCC, we have the technology transfer officer. So this office is also helping if you work with a new technology and you want to transform it to an industrial or for application. Yeah, I think the two most. Oh, the other thing which I feel I have to appreciate it, to have a mentor. It helped me so much. If you have a good mentor when you start your career, it makes things much easier for you.
Yeah, it's really important to have that support from the early stages of your research.
Yeah.
So thank you for that advice. And I guess as a concluding question, have you any other general advice for any students here progressing with their careers in STEM or pharmacy or whatever discipline they're from?
Okay, the research, I feel, if you like doing the research, UCC is a good place for research. They have a lot of help and support for the researcher. I think sometimes, after 1 year or 2 years from graduation, you have to decide, do you really like doing research, or it's better to go to industry? We see a lot of people who just keep staying long for research, and then they decide, no, it's not my place. They have to go to industry. Industrial experience make a huge difference for researcher, because you learn things which you will never learn in the lab. So if you get a chance to get intern or to have couple of months working in industry before doing research. This will help you so much. Yeah, I think that's all in my mind now.
That's really good advice. Thank you so much, Waleed. So if there's no other questions, I guess we will conclude there now for the evening. So on behalf of UCC Science Society and everyone here tonight, I would like to thank you sincerely very much, Waleed, for a very insightful evening. You have certainly succeeded in inspiring us all with your STEMinar, and we appreciate you taking the time to share your career and microneedle research with us.